Hello biology and welcome to your notes on DNA structure and replication. So you've heard about DNA, you know that it's the genetic material, and it was one of our 4 macromolecules. Today we're going to figure out what's with its shape, what's going on in there, and how do we get more of it when we need it. DNA is in the category nucleic acid, and that's our 4th macromolecule. There are 2 different flavors of nucleic acid. There's DNA, deoxyribonucleic acid. It gets a special name because the sugar inside of it is deoxyribose. The second flavor of nucleic acid is RNA. It's just plain old ribonucleic acid. So the sugar in RNA is ribose. Not too complicated of a naming method, but pretty hard to spell. What all nucleic acids, DNA and RNA, have in common is that they're made of these little building blocks. Just like carbohydrates are made of monosaccharides, nucleic acids are made of nucleotides. That's their monomer. It's the building block of any nucleic acid. There are 3 little parts, and some people like to say that it looks like a duck. Like this is the head of the duck if you gave it a beak, and this little pentagon is its body, and this is kind of his tail, so if that helps, it helps. The first part is sugar. This is color-coded, so that word's like purple and the pentagon is purple. That sugar can be deoxyribose if you are DNA, or ribose if you are RNA. The little red circle represents a phosphate. All we need to know about that is it's a little chemical group that definitely has the element phosphorus in it. If you remember, DNA is made of what elements? PCHON or pchon. That's where the P comes from, the phosphate. Here's where the N of pchon comes in. It's the nitrogen base. This is the duck's little tail. You see that there are a lot of different tails you could attach to that duck. Right now, the duck is wearing the adenine tail. It's a special kind of little chemical group. There are more. We have guanine, we have cytosine, then we've got the weirdos over here, thymine, only in DNA, and uracil, only in RNA. Subtle differences. Name all 4 nitrogen bases in DNA. You're correct if you said adenine, guanine, cytosine, and thymine. There's no uracil in DNA. Okay, now try it out for RNA. Name all 4. You're correct if you said adenine, guanine, and cytosine again, but this time leave out thymine and add uracil. Nucleotides, some of them are bigger, some of them are smaller. This is kind of a weird thing. So adenine and guanine, if you look in the bottom right, those are like the big boys in this diagram. You can also see it in this simpler cartoon version. Meanwhile, thymine and cytosine are like tinier, so is uracil. So the coolest thing about that is it makes your DNA the same width all the way down, because you're always going to pair like a big guy with a little guy. You're never going to have like 2 big nitrogen bases and like have like a lump in your ladder. That'd be super gross. The ladder's the same width all the way down because there are rules. The rules are A pairs with T and G pairs with C. Here's how you remember it though. Have you ever seen George Clooney? Have you ever? I'm going to Google image George Clooney so that you can look at him. George Clooney. Look at him. He's going to come up on the screen, I promise. There he is. Look at him. Look at George Clooney. And that's how you remember it. ATCG stands for look at George Clooney. Never forget. Now we're going to zoom out from like single monomers, and we are going to connect them all together into a really big polymer. So DNA is said to be shaped like a double helix. It's a fancy word for a twisted ladder. So the purple part of the double helix you see on the screen is called the backbone, and it's a sugar phosphate backbone that's really strong. So what 2 parts of the nucleotide are found in here? You're correct if you said that in the backbone, we are going to find deoxyribose and the phosphate group. In the middle, the rungs of the ladder are the nitrogen bases. They're held together by really, really weak hydrogen bonds. All you need to know about those bonds is that they're weak. They're so weak. What would be easier to break? The backbone or the rungs of the ladder? You're correct if you said the rungs of the ladder would be easier to break apart because they're held together by really weak hydrogen bonds. What am I pointing to, a hydrogen bond or a strong covalent bond? It's in between a sugar and a phosphate, and it's in the backbone. So this is a strong covalent bond, and there is a weak hydrogen bond between two whats? Between two nitrogen bases. We're throwing around a lot of words here. We have one more word to apply here, and it's anti-parallel. Hold on your hats, people. So if you have two lines that are parallel, they're running in the same direction and they're like equidistant from each other. So here are parallel lines. Anti-parallel lines are like whoosh. They're right next to each other, but they're running in opposite directions. So here's how that looks with DNA. I'm going to move myself a little bit. You can see the phosphate group on this left strand is at the top, but on the right strand, it's been like flipped over. So if you think of the phosphate group as the top, this left strand is standing straight up, but the right strand is upside down. Look at that phosphate down there. So they're not just parallel, they're running in opposite directions. So we call DNA an anti-parallel, running in different directions, double helix, a twisted ladder. This seems like a nitty-gritty detail, but it's actually really important for how we get more DNA without accidentally messing up your whole body with mutations. The other flavor, DNA's slightly less notorious little brother, is RNA. It has only a single strand most of the time, so it's kind of like a lonely, pointy ladder. Its base pairing rules are pretty much the same, except remember you gotta yeet thymine out of there and put who in thymine's place? Uracil. Adenines are going to be binding to uracils instead of thymines because there's no thymine in RNA. C's still bind with G's, that rule still applies. Look at George Clooney, always. Try this out. What types of nitrogen bases are in DNA? You're right if you said ATCG, look at George Clooney. Now change it up, give me the nitrogen bases in RNA. The only one that you should have switched out is switch out thymine for uracil. Next, we're going to dig into the individual nucleotides. Remember, nucleotides are made of what three parts? A phosphate group, a sugar, and a nitrogen base. So what sugar is in DNA? Hint, it's what the D stands for. In DNA, we have deoxyribose. And what is the R for in RNA? That is ribose. Last one, we just went over this part. What is DNA's shape called? You're right if you said it is an anti-parallel double helix. And last one, bring it home with what RNA is shaped like. You're right if you said it is a single strand. What I'm showing you right now is we're looking really, really close at DNA. Check out what we're looking at. Look at adenine and thymine, cytosine and guanine. Now look at the DNA twist together into a double helix. This is the level we're at right now. But we're learning about DNA because it makes us who we are. DNA is the instructions for making all of your proteins, and your proteins are you, baby. So when we coil up your DNA all the way and we squish it all together, you're going to get these familiar little things you've heard of called chromosomes that you inherit from your mom and your dad and that live inside all of your cells. I have a thinking question for you. I'm just not sure if you know this. Do you have all of the DNA in your tongue that you also have in your toe? Do those cells have the same DNA or different DNA? What do you think? Totally fine to be wrong on this one because I haven't told you yet, but the coolest part about your body is almost every single cell in your whole body has an exact copy of all of your DNA. So yes, your tongue cells have all the same DNA as your toe cells. They have cells full of the same chromosomes, which are made of the same teeny tiny DNA. This makes sense if you think about crime TV shows, right? Because they're always like, a hair was left at the crime scene. We can get a follicle cell from this man. And you can tell everything about them because all of their DNA is in that little skin cell on their head. The last thing I want to leave you with is, why does DNA's shape matter? How does the shape of DNA help it replicate itself accurately? When do you need more DNA? I don't know, you ever gotten a cut before? You ever got your ears pierced? If you lose cells and you need more, you're going to need to fill them with something. We need to fill them with DNA. They need the instructions for life, those cells. So we're going to take old DNA and use it to make the new stuff. Check it out. When a cell's ready to divide, maybe to grow, to heal a cut, it has to first copy all the instructions. The process of making an identical copy of DNA is called DNA replication. I want you to imagine that you have a book that tells you how to live your whole life. And before you have a kid, you need to hand copy that whole book, because when they're born, they're going to need the exact same one. So you better rewrite it and you better do it carefully. DNA replication makes sure that every single new cell has exactly the same DNA as the original. That stops us from getting mutations and even diseases like cancer that come from mutations. First, essentially your double helix kind of unzips. So if you look at the bottom, on the right you have an intact double helix that hasn't been unzipped yet. And here you can see an enzyme called helicase that is unzipping the chromosome. Then we're going to use the look at George Clooney rule. This is the coolest part. DNA is designed to be replicated. So once your two strands are unzipped, the whole ATCG rule makes it amazingly easy to add the correct partners to every single nitrogen base. So here you can see an old strand in yellow getting paired to new nucleotides in green, following the ATCG rules. And on the bottom, you could see the same thing. Here's the old strand in green on the bottom, and it's being paired with new nucleotides using those rules. It makes it really easy and really accurate. Lastly, when the whole thing is finished, your old strand has been peeled all the way apart, and you get two identical DNA molecules, except each one is actually half the old guy and half the new guy. We call this semi-conservative replication because you get two new double helices and every helix is half old stuff, half new stuff. I think it's a little easier to understand if you can see it in action. So the last thing we're going to do is watch this really cool animation of DNA replicating. So here we have the old strand. What do we need to do first? We have to unzip it. I'm going to change your life and tell you something that I never should allow to be recorded. It's the ultimate pickup line of high school biology, and it is it's about DNA helicase. You could get shirts that say this. I'm just quoting a shirt. It says, "I wish I was DNA helicase so I could unzip your genes. Look at DNA helicase. It's going to unzip the genes." Look, there he goes. He's breaking what kind of bond? Strong covalent or weak hydrogen? You're correct if you said DNA helicase is breaking weak hydrogen bonds. Is he breaking bonds between what part of the nucleotides? He's breaking bonds between the nitrogen bases. Let's keep going. Here you could see we're going to keep unzipping our DNA, and even as we're unzipping it, we are going to start using the base pairing rules. So here, let's click a little further. You're seeing how the old DNA is being paired with new DNA. All of these little boxes and circles and who's it's what's it's, those are just enzymes, and they're helping the process along. They're all different shapes to help us remember that enzymes are specific and they need to have specific active sites to get the job done. So up top, check it out. You have thymine and adenine, cytosine and guanine, thymine and adenine. We're following the base pairing rules so that we don't make any mistakes. What is this next thymine going to be paired with? You're correct if you said adenine. And what is this cytosine going to be paired with? Good one. It is guanine. And last one, what will this adenine be paired with? Be careful, it's DNA, so our answer is going to be thymine. If it was RNA, it would be uracil, right? Let's keep going until we get all the way to the end. The big picture takeaway here is that we ended up with two separate double helices, but they're like refurbished double helices because the top one is half old, half new, and the bottom one is also half old and half new. The beauty of DNA structure is it helps us replicate it by becoming like its own template for replication. If you just unzip it with helicase, you can use the @George Clooney rule to make a perfect full double helix. I hope that helped. I love DNA, so please come ask me questions about this. I'll see you guys later.